Welcome to the Week 3 question and answer session. Annie, did you have any questions?
I have 2 questions.
Okay.
My first question is, what is nitrogen starvation and how is autophagy linked to it?
Okay, so in biology, when we talk about nitrogen, we're generally talking about amino acids because nitrogen is the major component of amino acids. When cells don't have enough nitrogen, they run short of amino acids. And the outcome of that is that as amino acids are used to make proteins, the cell is not able to make enough proteins. That is really important because proteins form the structural and the functional components of cells. So if cells can't make proteins, then they're not able to grow and divide and do all those sorts of things, but over the longer term, they also can't survive. That's obviously a very serious problem for a cell. Autophagy provides a solution to that because it allows cells to bypass this shortage. And how does it do that? Well, it takes unnecessary or old or unused proteins, which the cell has, and it breaks them down again into individual amino acids, and it's then able to recycle them and use them for further synthesis, which allows them to adapt to the new condition and survive.
That really clarifies my question.
Okay.
My second question is there a mechanism for stopping autophagy?
That's quite a complicated question, but there is. In the vast majority of cells, there's a protein complex called TORC1, and that's a group of proteins which function together. And what they do is, their key role is in nutrient sensing. So when cells have access to enough nutrients, what TORC1 does is it is active and it goes around the cell modifying a whole lot of different proteins. One of those proteins is a protein called ATG13, which is the most upstream of all the different ATG proteins. And when ATG13 is modified, it isn't able to interact with all the other ATG proteins. That means that autophagy can't start because that interaction is essential for the initiation of autophagy. However, when cells are starved for nutrients, what happens is TORC1 becomes inactive, and that results in the modification of ATG13 disappearing. That means that ATG13 is able to interact with all the other ATG proteins and autophagy begins. In terms of stopping autophagy once it's been initiated, when cells are returned to a condition when they have plenty of nutrients, TORC1 immediately becomes activated again, and very quickly it's able to modify ATG13 once again, which means that autophagy is very quickly and effectively stopped.
Thank you very much. I think that clarifies all my questions.
Thank you.
Hi, my name's Hua. I'm really interested in the course. I have 2 questions for you, Alex. The first one is, what's the difference between autophagy in plants, animals, and yeast?
Okay, hi Hua. Thanks for the question. So there are some small differences between autophagy in yeasts and animals and plants, but I think it's more important to think about the similarities because autophagy is conserved between the 3. And if you look at the molecular details, so for example the autophagy proteins, you also see that there's a high degree of conservation in the autophagy proteins between animals and plants and yeast. And this says a lot about the important role that autophagy plays in cells because over a very long period of evolutionary time, autophagy has been retained between these different types of organisms. And that says obviously that it has a very important role to play in cells.
Oh, thank you. And the second question is autophagy helpful in cancer treatment?
Okay, so the link between autophagy and cancer is complicated. I'm not an authority on the topic, so I can't really speak with any authority. But I think that there are very promising treatments which are on the horizon. In some cases, autophagy is overregulated or it's more active in cancers. In other cases, it is defective in cancers. So there's a whole, it's very, very complicated situation. But I still think personally that it will be quite some time before we get a direct clinical outcome from autophagy research, simply because it takes a long time to develop clinical applications of basic research, and we're still at a fairly early stage in that process.
Okay, understand now. Thank you very much.
Thank you.